Okay, well, welcome everyone. Thank you for joining us today to this month's ISFE and Food Security and Nutrition webinar entitled Closing the Dietary Fiber Gap, Upcycling of Agri-Food Side Streams into Dietary Fiber Ingredients by Francisco Vilaplana. Francisco is Professor of Carbohydrate Biotechnology at KTH, Royal Institute of Technology in Stockholm in Sweden. He is originally a chemical engineer from the Polytechnical University of Valencia in Spain and holds a PhD in polymer technology from both UPV and KTH. After two postdoctoral stays in the Netherlands and Australia, he returned to Sweden in 2011, where he has developed his research activities in the academic and private sectors. Francisco has consolidated a broad research program focused on the exploitation of natural biomass resources through sustainable chemistry and enzymatic biocatalysis. The impact of his research focuses on two key areas of bioeconomy and sustainability: the development of lignocellulosic bio-based materials and the transformation of agri-food waste for functional and healthy nutritional applications. He has published over 120 scientific publications on diverse fields coupled to glycoscience, including plant science, bioprocess technology, material sciences, and food technology. He is also founding director of KTH Food, a multidisciplinary research center for a sustainable food system, and director of Plenty, a center for symbiotic and circular food provisioning. During 2021 to 2023, he was the chair of the review panel Chemical Engineering, Bioprocess Technology, and Environmental Technology at the Swedish Research Council, and during 2022 and 2024, he was part-time principal scientist at Oatly, where he led a research program on plant structure and health. Now, before we start, please remember to mute your microphone during the presentation. If you have any questions through it, you can write them on the chat so that you so that you do not forget them. And please mute your microphones. So to ask the questions at the end so that you do not forget them, or at the end you can ask them directly to our speaker, or I can also read them out loud for you. So again, thank you so much, Francisco, for this time. It's a pleasure to have you in here. I'll mute the microphones. Thanks, Francisco. Now the audience is yours.
Well, good morning, everyone, or good afternoon, depending on where you are. First of all, thank you very much, Viridiana and Professor Jorge Belti, for the very kind invitation. For me, it's a real pleasure to be with you today in this seminar. So yeah, the idea would be to show you some of the research that we're doing in my group, but also to introduce you the field of food at KTH and also the Plenty Center that Viridiana mentioned. So I'm going to share now my slides for you. Let me see if everything works fine. It looks like it. You can see the presentation, I assume.
Yes.
So anyway, well, thank you, Viridiana. You already mentioned all my background. Exactly. I've made actually a personal research transition. I started as a chemical engineer, then I did my PhD in material sciences. But I really want to mention that it was during my postdoctoral stay at the University of Queensland, where basically I fell in love with food, mainly with carbohydrates, starch, and dietary fibers. And that has accompanied me through my research journey. What we do in my group is quite broad. Our main competence is carbohydrate biochemistry, but we look at the structure of the different carbohydrates in different biomass streams from the plant cell wall, but also from the fungal cell wall. And there we collaborate with plant and fungal biologists. And then we utilize this fundamental information about the structure and the interactions of carbohydrates in different biological tissues to design different processes using green chemistry and enzyme technology. And we are mainly focused in two main areas of the bioeconomy. One is what we call bio-based materials, and this is very important for Sweden. Sweden is almost 50% covered by forest. So basically half of my research group is developing new wood-based materials that could replace for example, fossil-based plastics or other types of materials. But the other half of the group is still working on food and nutrition science. So this is what I will talk to you today in this seminar. But I also wanted to introduce a little bit the food sector in Sweden. Maybe you don't know Sweden for the food, obviously. I mean, maybe it's known for other things by IKEA or Ericsson or Spotify. But actually, indeed, the food sector in Sweden is the third largest industry. There you have some big numbers about it. And I actually want to highlight here that, I mean, Sweden is very well known for innovation and technology, but unfortunately, the innovation in Sweden in the food sector, I believe, and that's something that we're really trying to develop, is to increase the value, because it's basically lagging compared to other countries in Europe. And with this background, we established KTH Food, which is an internal center at our university. I think it has a very similar idea than of your food security and nutrition program at Tec de Monterrey, which I think is a fantastic initiative. So we also try to integrate the different competencies that we have in 4 main areas: sustainable food production, circular food processes, value chain and consumption, and functional food products and health. And I mean, at some opportunity, you're really interested to collaborate in any of these topics, you can, of course, contact me, and I will direct you with the different wide array of researchers that we have at my university in Stockholm in this area. But today, I'm wanting to talk about one of the food components, one of the important ones, mainly the dietary fibers. So we're all very well aware that the structure of food is very important to its health. We know that our diets have evolved from a complex plant-based diet with a slow digestion and prolonged release of nutrients to a more westernized diet, which is structurally simple, with easy digestion and fast release. And one of the main issues here is basically the low consumption of dietary fibers that we have in our diets. And this is what I wanted to introduce you. This is one of the challenges that my group is trying to address, is exactly what we call the dietary fiber gap. And on the right side, let me just put here the pointer. Well, now it doesn't work. Here we go. So here you can see basically what we call the dietary fiber gap is the difference between the actual consumed amount of dietary fibers and what is recommended. And I'm actually very happy to see that Mexico actually lies quite high on the top. So you're doing pretty good, but as you can see, most of the Western countries actually, they do not eat as much dietary fiber as it is recommended. Basically, we know there are different dietary recommendations from different countries, but in Sweden, the Nordic Nutrition Recommendation from 2023 mentioned that we should consume between 25 and 35 grams of dietary fiber per day. And as I said, we are lagging behind in most societies. But what is dietary fiber? The concept was initially coined in the 1950s, and this concept has basically evolved throughout the years. And here I really want to spend some time with the definition, because I think it's very important to understand also the different technological approaches that we need to achieve to increase our consumption of dietary fiber. So this is the definition that was agreed in 2009 by the Codex Alimentarius Commission. And basically, dietary fiber is any carbohydrate polymer, and here it's very important to go down on the footnote, or lignin. So it's also associated compounds to carbohydrate polymers are included with a specific size. And in 2009, the Codex Alimentarius recommended 10 or more sugar units in the dietary fiber, but you can see there's another footnote down there. And in some countries, actually, oligosaccharides between 3 and 9 sugars can be included in the definition of dietary fibers. And that's particularly the case in Europe and also in the United States. But anyway, dietary fibers are these carbohydrate polymers and oligosaccharides and associated molecules that cannot be hydrolyzed by our enzymes in the small intestine, and then basically belong to one of the different categories here. So they are edible carbohydrate polymers that are intrinsically and natively in the food that we consume. Then a second category is carbohydrate polymers that are basically isolated from these food raw materials by physical, enzymatic, and chemical means. And this is very important for these isolated carbohydrates, you need to demonstrate a physiological benefit to the health. So this is very important because as those of you working in nutritional benefits of different food ingredients, to really have an associated physiological benefit and to demonstrate it normally is quite challenging. And then the third category, as you can see here, is synthetic carbohydrate polymers that again need to show a physiological benefit to health according to the different scientific evidence. So this is what we are working with. Classification of dietary fibers. There are different ways of doing that. This is one of them. You can classify them based on the size. You have high molecular weight dietary fibers, mainly non-starch cell wall polysaccharides like cellulose, hemicellulose, pectins, or gums. We have resistant starches, which are the type of starch that cannot be digested again by our enzymes, either by chemical or by physical hindrances. And then these associated substances that I was mentioning before, for example, lignin, that can be present in the cell wall of different plant ingredients, plant raw materials, or cutin or suberin that you can find them, for example, in tomatoes and other vegetables. And then you have, of course, these low molecular weight oligosaccharides, very familiar, fructooligosaccharides, galactoligosaccharides, xyloligosaccharides, inulin, that belong to this category. But then we can also classify dietary fibers based on the functionality, and then probably you've heard the definition of soluble, insoluble dietary fibers, or fermentable, non-fermentable dietary fibers, or viscous, non-viscous dietary fibers. But this definition on functionality can be a bit difficult because, of course, it's very difficult to assess solubility. It's very difficult to assess fermentability. It depends very much on the environment and how basically these dietary fibers are presented. So what I really like is basically to normally explain the chemical diversity of dietary fibers. And here we always need to go down to the basic biochemistry of carbohydrates, maybe from the first year of university in biochemistry. So this huge diversity of dietary fibers is what we are dealing with and that we are working on. And then that diversity of dietary fiber depends on the constituent monosaccharides, on the anomericity of the glycosidic linkages, if it is alpha or beta, the connectivity of the glycosidic linkage, the degree of polymerization, and the type of macromolecular topology. And this is very important for us because when you read some of the nutritional studies on the dietary fibers, they don't go into the details of this huge diversity of biochemical structures. Maybe they just consider dietary fiber as one category. But this is exactly the wide diversity of biochemical complexity that we're dealing with when we're exploring dietary fibers. But not only that, because dietary fibers, as I was mentioning, they can be isolated, but they can also be present in very complex native and intrinsic assemblies. And that is correlated to the macromolecular, supramolecular architecture of the different types of foods that we're dealing with. So when you look at the cell walls of for example, cereals or fruits, of algae or mushrooms, you're always encountering, a wide diversity of macromolecular components. You can have cellulose, you can have arabinoxylan, proteins, lignin, and they are not basically just mixed. So these components actually are interlinked by different covalent and non-covalent interactions, and that really contributes to the difficulty or the accessibility of these dietary fibers when they reach the colon. They can also form ordered crystalline, for example, or amorphous conformations. And you can see that they actually form very complex multi-scale architectures from the nanoscale or nanometer scale, when you will have, for example, individual cell wall microfibrils, to even millimeter range when you're looking, for example, at the different tissues in cereal cell walls. And this complexity is normally very well organized and very well designed by the biosynthesis in the different organisms. But what are the beneficial health effects of dietary fibers? So we always need to differentiate them depending on when they occur. So some of these beneficial health effects, they actually already occur in the upper gastrointestinal tract. So before they reach the large intestine, and that is basically due to their physicochemical properties. So dietary fibers as carbohydrate polymers, they are hygroscopic. They can attract water and cause swelling when we eat our foods. And this is directly correlated to a reduction in satiety. Also, in a similar way, they can increase the transit time and modulate the nutrient uptake in the small intestine. And for example, they can contribute to a reduced release of glucose and lipids, which is something that we are also looking into some food that have a prolonged release of energy. And also, in some cases, like in the case of mixed linkage beta-glucans from cereals, they are also related to a reduction of cholesterol levels, specifically due to this viscosity that beta-glucans have. But that's not only it. Of course, when they reach the large intestine, then they have a very positive effect on the gut microbiome. And that is what we call a prebiotic effect. And this is because these dietary fibers, they indeed become the main carbon source for fermentation of our gut bacteria. So basically, as I was mentioning, they remain intact throughout the upper gastrointestinal tract. They reach the large intestine, and then they become the carbon source for fermentation of our gut bacteria. And what is really interesting is based on this diversity of chemical structures of the dietary fiber, then it will determine which bacterial communities they can basically be utilized for. And then these dietary fibers, when they are fermented, they can release important bacterial metabolites. Maybe you've heard of short-chain fatty acids that are very important and very important bioactive compounds with positive health effects. And they're also involved in improving the barrier integrity of our colon cells and also involved in reducing inflammation. So you can see that this is a very important cascade effect. And this is basically, again, why doesn't it work? This is basically depicts the same way. This is what happens to the dietary fibers. Then they will reach our large intestine. You can see here a very interesting cartoon of what a cell wall of a cereal could look like. And then you can see how our different gut bacteria, they can release some of these bacterial enzymes that will be able to hydrolyze some of the dietary fibers that reach our large intestine, break them down into smaller oligosaccharides here that then can be transported inside of the bacterial cells and be further metabolized into the smaller monosaccharides, and then, as I said, produce bacterial metabolites, again, these short-chain fatty acids that have very important functions in the whole gut homeostasis. So they can be utilized by our colon cells as carbon source, like, for example, propionic acid, and then they also have other important health benefits. But what is very important to understand is, of course, the complex bacterial machinery that every bacteria type will have is what determines whether a dietary fiber can be utilized or not by a specific bacteria. And based on that genetic machinery, different bacteria families will encode the specific carbohydrate active enzymes that will be able to utilize different types of dietary fiber. But that's not only it, because we have a huge ecosystem of bacterial species in our gut, and then sometimes these dietary fiber metabolisms cannot be only considered as only from the perspective of one bacteria, because it's a community effort, and in some cases you have competitive or cooperative efforts, and all this complexity is what basically determines the functionality of dietary fibers. And I just wanted to highlight here, going a little bit more into the biochemical complexity, is that even for one type of dietary fiber, and I present here the case of arabinoxylan, which is one of my favorite polysaccharides, or I would say the one that I've worked most throughout my career, depending on the source, on the plant source of this arabinoxylan, if it comes from wheat or from rice or from corn or even from other species like spruce, etc., the molecular structure is basically different. They share some features, but the fine structure is different in the types of sugars, the types of linkages, the type of polymeric architecture, and also the wide range of enzymes that is needed to be able to deconstruct these dietary fibers would be then very different depending on the type of fiber. So here, for example, if you would need to deconstruct, this is a picture of corn arabinoxylan, which is one of the most complex dietary fibers that we have. We will need the concerted action of around 10 to 12 carbohydrate active enzymes in able to be able to break down fully this complex polysaccharide into the constituent monosaccharides. So you will need enzymes that will cleave the backbone, for example, xylanases. You will need enzymes that start chopping the side chains, glucosidases, xylosidases, galactosidases, et cetera, et cetera. So you can imagine, the wide complexity of dietary fiber metabolism. And not only that, so for each type of enzyme, there is different types of of different types of families depending on the way they hydrolyze. So here is an example, the type of the xylanase family, which is an enzyme that specifically cleaves the xylan backbone in specific positions, depending on the type of glycosyl hydrolase family that you have. So basically, depending on the structure of the protein, of the enzyme protein, it will actually act in different ways. So this is a cartoon, for example, of the two main families of glycosyl hydrolases, xylanase family 10 and family 11. They actually will cut the polysaccharide in different positions, depending on their ability to accommodate different side chains. But also there are very more specific xylanases, like this specific xylanase from the family GH30 that specifically requires the presence of a glucuronic acid in order to cleave here in this position. And similarly, a glycosyl hydrolase family 5 xylanase that requires an arabinose. So you can see that it's not only about the functionality of the enzyme, it's also the different families will have different catalytic effect in our dietary fibers. And that basically brings us to basically one of the main questions that we want to answer in my research is that how does the biochemical and biophysical structure of the dietary fibers influence their fermentability in the colon? And most importantly, from a functional point of view, can we specifically design dietary fibers that would target specific bacteria at specific points in our colon? And previous research by other groups in the US definitely demonstrate that it is possible to basically combine different fibers based on their specificity to different types of bacteria. So you have fibers that show, for example, low specificity, meaning that a wide range of bacterial species could be able to utilize them. But then you have other specific fibers that only niche bacteria could utilize. And basically what we want in our diets is a mixture of low specificity and high specificity bacteria. And in the end, if we would like to increase dietary fiber content in food products, it is also very important to understand that not only technological developments are needed. And here, for example, I'm talking about, efforts from plant biology. So how you can, for example, increase the amount of dietary fibers in different plants by breeding processes, or you can increase it for processing techniques or fortification. But you also need to make sure that these enriched dietary fiber products have actually very beneficial functional and sensory properties, because most of these dietary fibers actually have viscosity, they have taste, they might have solubility issues. So we really need to make sure that, that the addition of dietary fibers basically impact the functional properties in the way that we want them. But that's not only all enough. We need to make sure that consumer acceptance is there. So for example, the impact of gut health. Now, consumers are more worried about, the effect of different foods into our gut health, personalized nutrition. This needs to be taken into account. And finally, of course, regulatory acceptance, because in different geographical areas, you need to consider if a specific new food product with high fiber will be considered as novel food. It also has an impact on whether the process would change the definition of dietary fiber, health claims, et cetera, et cetera. So it's not as easy as it sounds. It's basically you need to align these different aspects. But after this introduction, I really wanted to go a little bit deeper into some of the research that we're doing in my group and connect it to Plenty. So what we are actually dealing with is what I call the other type of fiber gap, which is based on the fact that 1/3 of the food that we produce in our systems is lost and waste. And basically, this contributes to 10% of global greenhouse gas emissions. But most of these side streams and processing waste are really rich in insoluble fibers that we are not currently using from human consumption. And we all know, we're very well aware of, when we make flour or white flour to make bread, most of the dietary fiber actually ends up in the bran layers of the cereals, and we give it to animal feed. Or the same when we make, for example, fruit juices. When we press the fruits, we get our wonderful juice, but then most of the fiber actually ends up in the pomace that normally ends up as either animal feed, or goes to biogas production. So that's exactly what we're targeting in this center, Plenty, that I am coordinating at KTH. It's a center for symbiotic and circular food provisioning, where basically our vision is to change the way we look at side streams and waste across the food system and try to reintegrate them back again into the food production system. We are looking at different work packages. We're looking at different perspectives. Of course, the technological approaches have to be there. We're also looking at the systems perspective. We're looking at the societal benefits, and we're also looking at the innovation management. So for the valorization of the different agricultural losses and side streams, of course, you can use different technologies. We can be using extraction technologies, fermentation technologies, enzyme technologies, thermophysical processing, and chemical processing technologies. And basically, the idea of this center is to try to optimize the best technology adapted to the different side streams that we have, and also targeting into the different products that we're getting. And we're always trying to go up in this pyramid of waste use, trying to go up from biogas production or to production of food and feed, which is in the end what is our main goal. So what we're doing in my research group, basically we are looking at two main technologies. One is extraction technologies, or using green solvents. And here we've been using for many years, for over 10 years, a technology called critical water extraction, which basically means using water at high pressure and high temperature, basically keeping it in liquid phase at high temperatures. And that allows us to solubilize dietary fibers from insoluble side streams, like, for example, from cereals, from fruits, etc., basically with high molecular weight, and also preserving the native structures. And I will give you some examples of our results in the coming slides. And the second approach that we're doing in my group is enzyme technology. And here we really want to utilize the exquisite selectivity of carbohydrate active enzymes for specific high-value applications. And that's normally directed for the production of prebiotic oligosaccharides with specific molecular structures, but also to functionalize dietary fibers towards, for example, increased viscosity or increased rheological properties. And I will also give you some examples of these results in the coming slides. But as I said, first of all, we really look at our cereal side streams as sources of new food ingredients. So this is basically, you're very familiar with this. This is the cereal grain. You have the endosperm region. That's where the main of the starch and proteins are. But then you have these thick walls of the bran. And you here have a picture that we took in my group with using tomography of a bran layer. And here you can see very nicely some of the residual starch granules. You can see here the aleurone layer, and you have here the pericarp layer. What is very interesting is, of course, that these bran layers are very rich in dietary fibers, again, and insoluble dietary fibers. And this insolubility actually comes from a very complex molecular architecture of these cereal cell walls. So here is a cartoon of the cereal cell wall. The main component is normally arabinoxylan again. So you have the xylose backbone, you have these wonderful arabinose substitutions, and these arabinos are actually covalently bound by what we call diferulic acid dimers. And these are actually not sugar components. They are phenolic compounds. And what they do is basically to make these insoluble, to make them cross-linked. But we not only have that, we have lignin, we have proteins, we have cellulose, we have beta-glucans, we have pectins. So basically this complex architecture is what we have to deal with when we're dealing with different insoluble native and intrinsic dietary fibers from cereals. So basically, the solubilization of dietary fibers from cereal cell walls, most of the process, or traditionally, people have extracted these dietary fibers using, for example, alkaline extraction. This works very well. The issue is actually that you break these covalent linkages, these ester linkages between the arabinose and the ferulic acid. And unfortunately, we lose this compound ferulic acid, which has demonstrated antioxidant and health benefits. So what we're doing in my group, and we have been working with this technology for many years, patented technology, is to use subcritical water extraction, where we actually are able to isolate these arabinose islands in polymeric form, high molecular weight, but also preserving around 10 to 50 or 1 to 5% of this ferulic acid content in the soluble dietary fibers. And we also demonstrated that they have radical scavenging and antioxidant properties, which we think that they are very, very important. So we have extended this technology to different biomass sources. We have used it for example, wheat bran, rye bran, barley bran, corn fiber. And what is very interesting about subcritical water is that controlling the temperature, controlling the pH of the water, controlling the time of the structure, we can really fine-tune the molecular architecture of the dietary fibers that we are isolating. So you can see here that this is a very clear depiction. So basically, by fixing the pH, we can preserve the molecular weight of the arabinose islands at polymeric form. When we are not buffering the system, we can actually tune the molecular weight reaching smaller sizes. So this is actually quite relevant when when you really want to target the functional properties of the dietary fibers. We basically have applied this technology not only to cereals, but also to fruits, to mushrooms, and to algae, and it works very nicely in all cases. So this is an example of a similar study that we did subcritical water extraction of pectins from apple pomace. And again, very similar story. Basically tuning the pH and the temperature of the subcritical water extraction, we can enrich different parts of the pectin molecular structure. So if we want to have a dietary fiber with high homogalacturonan content, then we need to decrease the temperature and the time of extraction. So normally homogalacturonan regions are easily extracted, but if we would like to increase the amount of complex rhamnogalacturonan 1, then we need to increase the temperature and time of extractions, and then we will be isolating these branched parts of the pectin. And this is the same for the degree of methylation. So the degree of methylation of pectin is a very important functional property of the pectins. So if we would really like to have a low methylated pectin, we will need to use acidic conditions. If we want to have a high methylated pectin, then we will need to use neutral conditions. And that, in the end, allows us to tune what are the functional properties of the ingredients in this particular case. So we could demonstrate that depending on the conditions of the extraction and the molecular structure of the pectins, we can have different types of gels with specific rheological properties. You can see here that our gel at 120 degrees and pH 7 had very nice rheological properties, while also if you want to use them for emulsifying properties, a pectin with high degree of methylation would be beneficial compared to a low methylated pectin. So this is a very nice showcase of how really you can tune the conditions to get a specific structure, to get a specific properties. And now looking a little bit more into the enzyme side. So we are using different types of carbohydrate active enzymes. We're using mainly glycosyl hydrolases, that are the enzymes that will hydrolyze glycosidic linkage. We're also using carbohydrate esterases that are active on ester bonds, specifically between carbohydrates and other non-carbohydrate substances. And we also use what we call auxiliary enzymes, that are basically enzymes capable of oxidizing different polysaccharide components. And we use these enzymes depending on what type of application we want to use. So I just wanted to, as an example, highlight you this project that we had in collaboration with Novozymes in Denmark, now called Novonesis. It's one of the largest enzyme providing companies in the world. And together with them, we really want to explore the use of different xylanases of different families. I already introduced them to you before. from family 10, from family 11, from family 8, and from family 5 that basically will specifically cleave the xylem backbone at specific regions. And based on that, we could get a specific oligosaccharide profiles. And this is a very good example. Like, for example, from using a xylanase from family 11, you mainly get oligosaccharide profiles that are very small in size. You will only get xylose and xylobiose. And this is not very good for the dietary fiber definition because you can see here that very small oligosaccharides of degree of polymerization 1 and 2, this will classify as sugar and not as dietary fiber. Like, on the contrary, by using an enzyme from the family GH8, you will get a much more diverse profile of oligosaccharides. We'll still get xylobiose with two xylose units linked, but you will also get xylotriose, xylotetraose, pentose, and much larger oligosaccharides. So these will fall into the definition of dietary fibers. And most importantly, that also will influence the growth of selected gut bacteria. And we could demonstrate that the profile selected by the family 8 basically would increase the growth of specific gut bacteria. So this is very important to be able to fine-tune the enzymes, the size of the oligosaccharides, and then the prebiotic effect of the oligosaccharides. And then we're very interested as well in digging into a specific family of xylanases, which is from the family 5. I already mentioned it. This is a very interesting family because it requires an arabinose to cleave. And long story short, we get very long oligosaccharides from these families, and that means that most of these oligosaccharides will be counted as dietary fiber and will really increase the solubility of the arabinose island. So this is something that we're exploring in collaboration with Novonesis and with Arla to be able to release as much dietary fibers as possible from these insoluble side streams. And another example is the use of ferruloyl esterases. These enzymes are not hydrolytic. They're capable of cleaving the ester bond between the arabinose and the ferulic acid. And we're exploring them in collaboration with different xylanase families. And basically, long story short, we can demonstrate that having these ferruloyl esterases together with xylanase, we increase the amount of oligosaccharides that are being released from these insoluble side streams, and tuning the amount of tuning the family of ferruloyl esterases also has an impact on the products that we are releasing. And in the last 5 minutes of the presentation, I just wanted to show you some very few examples where we can use a specific cross-linking enzyme, specifically lactases, to produce hydrogels from wheat bran. And from corn bran, you can see here, we've been working with wheat bran and arabinose island, which has a backbone, which has a low degree of substitution, a ferulic acid content of 10 milligrams per gram. And here you can find corn fiber that has a different structure, much bran structure, higher content of ferulic acid, and lower molecular weight. So we're using these enzymes called lactases that are oxidative enzymes. What they do is basically, in the presence of oxygen, they oxidize the ferulic acid, they create a radical, and basically through radical coupling, we can make these covalent bridges that basically reconstruct the original covalent bonds that we find in the cell walls. And what is very interesting from a functional perspective is that here you can see the viscosity curves for the specific fibers. So for the soluble arabinose island, we have a low viscosity. It behaves basically like water. When we introduce the lactase reaction and we do the covalent cross-linking, we increase the viscosity. It behaves like basically honey. But when we dry and we regenerate them in water, basically we create textures like, for example, I would say it, like collagen, so very thick rheological properties. You can see here, like gelatin. That was what I wanted to say. So you can fine tune the rheological properties of the hydrogel based on the reaction and the way you handle it. And this is basically, I'm not going to go into details, but this is basically due to the way that these fibers are assembled. It's both due to the covalent crosslinks, but also due to the backbone interactions. And we're trying to even explore this concept by using different other enzymes to be able to fine-tune the rheological properties using also hydrolases. And I just wanted to mention that this is actually very important from a health perspective, because this is a study that we did using in vitro cell cultures. So what we did was basically to put in vitro colon cells in contact with our hydrogels that have this ferulic acid, and we stress them with specific radicals. And then you can see here that when these cells were in contact with alginate, basically this, you can see a fluorescent intensity coming from the radicals, but when they were in the presence of our antioxidant dietary fibers, basically the fluorescence decreased, basically meaning that these radicals were quenched by our dietary fibers. So you can actually imagine now we're trying to explore this concept in vivo, in different mice models exposed to inflammation, and we really hope that these antioxidant dietary fibers really can have an effect on preventing inflammation in our colon. And now we're exploring this concept, trying to basically reconstruct these native dietary fibers by introducing, for example, cellulose microfibrils, basically in order to increase the complexity of our cell walls, to couple them with also proteins, to couple them with phenolic compounds, basically with the idea to reconstruct the cell wall. And just as an example, I wanted to mention the work that we're doing in order to improve the texture of plant proteins. So we all know that replacing meat proteins by plant-based alternatives, it's a very big scientific and societal challenge. And we know that some of these plant proteins, unfortunately, do not have good textural properties. So our approach is that combining these plant proteins with some dietary fibers, we can actually improve the texture, improve the rheological properties of the dietary fibers. And we have basically demonstrated that for gluten proteins, for pea proteins, and for soya proteins, basically combining them with dietary fibers, we can again tune the rheological properties of these plant proteins. And just wanted to maybe finish here, because just wanted to basically convey the main message of my presentation is that, dietary fibers are complex and heterogeneous family of biomacromolecules with a wide range of molecular structures and topologies, and that's very important to understand and to control when we want to use them based in functional food products. The second main take-home message is that the cell walls from plants and mushrooms and algae are a rich source of intrinsic dietary fibers, and this is important. Unfortunately, some of these insoluble cell walls don't have positive taste or positive functional properties, and that's what we want to utilize in my group. We want to convert these insoluble and non-functional dietary fibers into functional ones that we can use in specific food products. The next take-home message is that both the biochemical diversity and the physical accessibility of the dietary fibers will determine the way that they are functionalized and utilized by the gut microbiome. And this is basically what we will define as prebiotic dietary fibers. And then this is one of the scientific questions that we're trying to address in my group. So is there a hierarchy of dietary fibers based on their specificity, going from broad dietary fibers that can be utilized by a broad range of bacterial populations in our gut, and others that are more selective and more specific to specific families or even phyla of bacteria? And in the end, the main question is, can we design dietary fibers that have both specific functional properties, but also targeted fermentability in our colon? So this is basically the main things that we are addressing. And I just wanted to take the opportunity to really thank mainly Cecile and Rescandi, who were the PhD students that were involved in the preliminary work that I have shown on arabinose island utilization. I want to thank my colleagues from Novozymes and from Landmannen that have been very fundamental in specific parts of the project. And I also wanted to mention specifically what is, for example, Carl's project that we are now trying to expand this approach to encapsulate different specific bioactive compounds to be able to deliver them specifically to the gut, and of course the rest of my team at KTH. And I will be very happy to answer any questions from you. Thank you very much.
Thanks so much, Francisco, for this knowledge that you just shared with us. Now let's start the question and answer portion of this webinar. Remember to turn on your microphone or raise your hand so that we can unmute the microphone so that you can ask the question directly to our speaker, or you can also write them on the chat and I can ask him for you. Any questions? Aurea, let me see. Can you unmute yourself? No.
No.
There you
yeah.
are.
Yeah, thank you very much, Francisco, for this amazing presentation. I want to ask you, as you might know, in Mexico, we have some interesting insoluble fibers. For example, you know that I work with agave, with mezcal, and these fibers that are discarded after the distillation process to obtain mezcal or tequila. I think it's a interesting source of insoluble fibers. However, we know that we cannot use these fibers directly for human consumption. So how can we apply glycoengineering to design food products suitable for human consumption, either directly or through other processes, for example, fermentation, precision fermentation to obtain metabolites, single-cell proteins, or other type of compounds?
That is a very good question. Thank you, Aurea. No, I think this is a very relevant question, and I think the strategy can be applied basically to any fiber. Sorry. What I will start always is try to understand what is the composition and what is the molecular structure of these dietary fibers that you have in the agave. I think we looked at it. Probably there will be a lot of pectins there. Probably there will be a lot of cellulose there. And then based also on the applications that you want to get in the end, if you want to go for fermentation technologies to a specific metabolite, then of course you need to engineer. You need to engineer the whole process. It's like, do you want to deconstruct all the dietary fibers as carbon source for the fermentation, or do you want to use the dietary fibers as such? For example, maybe get them isolated in polymeric form so you can use them directly. So I think it depends so much on what a procedure you want to, or basically what technology you want to use and what end product you want to use. But I think it all starts trying to understand as much as possible the native structure of the dietary fibers in the agave. But I'm really happy. We've discussed about this before, so I will be very happy to explore potential applications or technologies that could be even complementary.
Yeah, of course. Well, thank you, Francisco.
Thanks, Aurea. Anyone else? I do have a couple, if no one has. Okay, so thanks, Francisco. We've worked a lot also with byproducts from fruit peels. Particularly myself, I've worked with extrusion and high pressure processing to transform the solubility of dietary fiber from insoluble mainly to soluble based on just the processing, which is very interesting. We've worked also with Professor Hamaker at Purdue University with these in vitro fecal fermentations and seeing what could we do with the same matrix, but just transform it by using any process that we are experts on. So to see this is like very exciting to see that someone is keeps on going with this kind of work. So congratulations for that. My question was, well, that was a comment. My question is, now we're working with insects. In kiting, it's been kind of a difficult molecule to understand. Do you think with what you've done previously that it can be actually considered as dietary fiber, and can the current methodologies that we are using, like the ones you described, or the ones that we have from Megazyme, for example, are those ones capable to actually quantify chitin as dietary fiber?
Very good question. So I will first reply to your comment. I'm really happy that you're working with Bruce Hamaker. I've known Bruce from my postdoc times when I was in Australia 15 years ago, and I'm always inspired by the work that he does. So So yeah, I think we all share the same passion for dietary fiber structure and application. So I think we should really build a community, because as you know, there's a lot of talk about proteins, protein, but what about the fiber? I think hopefully we can all, talk together and work together to really increase the profile of dietary fibers. So that makes me very happy to hear. I haven't seen Bruce in some years now, but I hope he's doing really well. Now going to the kiting question. So two things. So I think based on basically the methodologies that Megazyme has for dietary fiber, chitin already would be included as such if it is present in intact food products, if you understand what I'm saying. So if you're already, for example, eating a mushroom that has chitin also, that chitin counts as dietary fiber. The issue is, as you know, this is a very difficult polymer to work with. It's insoluble. It's very similar to cellulose. However, there are enzyme technologies that can break down chitin. There are chitin lytic polysaccharide monoxygenase. It's a specific type of enzyme. I am not working with it personally, but that's something that we could explore together. But there are catalytic enzymes, chitinases, that work with chitin. So this is actually a very interesting challenge, both from insects, but also from other sources like mushrooms, for example, or even shellfish. So I would say that, yes, most of the technologies that I have explained, they can be used for chitin, but of course you need to adapt them and to look at the specific details.
Thank you so
That
much.
That was a long answer.
No, that's perfect. Thank you so much. Orlando, please. I also do have a couple in the chat, so I'll ask them after Orlando.
Thank you so much. Dr. Francisco, thank you so much for the presentation. I think it was very, very interesting. I work with oligosaccharides, so it was very interesting for me too. And with valorization of some agroindustrial byproducts. My question was, you show one slide that you assess the prebiotic activity of different degree of polymerization of Hillose polysaccharides, right? My question is if you have assessed the prebiotic activity based on cross-feeding of different communities on the different dietary fibers based on the source, because the source varies very much on the targeted community that it can impact. So it's very interesting for me.
Give me a second. You should be able to unmute yourself. There you go.
Yeah, sorry, I muted myself because I have a bit of a cold. I was coughing. Anyway, so thank you, Orlando, for your question. I think this is the key. I think that's the main question to address. No, we haven't. Let me see, how can I say this? Yes, we have studied the effect of different let's say, xylem topologies in complex fecal fermentations. I didn't present the data because it's not published yet, but what I can tell you is that what we have discovered is that it's not so much about which type of bacteria you have. So the diversity of the bacteria is about the functionality of the bacteria. It's about what machinery the bacteria has that allows to utilize a specific type of fiber based on the source. And that's a little bit what I tried to explain in that slide when I had xylem of different sources with different structures.
Yes.
So basically, let's take the most extreme cases. Corn xylem is very complex, but with arabinoxylem is not so much. So of course, they will, depending on the structure, they will target different populations, and some of them will work together to be able to deconstruct them, because some of them have the enzymes and others have other enzymes, but other species will actually compete.
Yes.
And and then basically you need to study all of these processes together, because as I say, this is an ecosystem. You can think of this could be considered ecology project, because you have all these different species with different catalytic processes, etc., etc. So it's a very complex situation. But what we discovered in this study is that it's the functionality of the bacteria more than the diversity that seems to play a role. I hope I
Thank you
answered.
so much. Yes, yes. That's exactly what I was wondering, and I was wondering if you have immersed into that. So I hope to see the results you are about to publish
Yeah, me
soon.
too. It's taking forever, but hopefully we'll get there somewhere sometime.
Thanks,
Thank you so much.
Orlando. I'll ask one last question because we're running out of time from Alejandro, which is in line for the questions. So how can computer systems engineering contribute to these investigations?
Yeah, this is a very good question. This is something that we're working on. I am not a machine learning person, but of course now it's in there. So one big example is basically AlphaFold. I mean, you all know of AlphaFold. That's basically machine learning or computer systems works in a specific protein design. But you can think in the same way at a bigger process system. If you really want to, if we could categorize dietary fibers based on structure, and that will be an input for the computer system. Then you can maybe have the process in between, test different applications, and then get in the end, which products are you getting. So this is another way of looking at it. But definitely, this is something that I think we are all aware that this is the future. It can be applied to basically any field. Maybe you're looking at extrusion processes also assisted by artificial intelligence, where you can basically tune the properties of the extruder to get a specific textures. I mean, I think to everything.
Well, maybe 1 minute for one last question. Is that okay with you?
Yeah.
Given its functional, this is from Mariana Martinez, given its functional versatility and GRAS status, what do you see as the most innovative and underexplored applications of cellulose in food formulation that could reshape the future of clean label product development?
That very, very good question as well. Let me see how I respond this. I haven't worked with cellulose modification myself, but we already know some of the processes that are there. One thing that actually I believe, and actually when you mentioned it, I think it was you, Viridiana, I'm very interested in high pressure processes, and specifically for cellulose functionalization. We are working with this not on the food area in my group, but on the wood area of my group. But again, I think it has a lot of potential to be able to work with very complex crystalline cellulose. So this is something that maybe something for discussion that I would be very interested in working together, because then you don't need to extract. You see my slides. We try to extract and get the soluble fibers out, but maybe with these high pressure techniques, maybe you need to. It helps basically with the dispersion and the functionality. So this is something that sounds quite promising to my ears, basically.
Well, thank you so much, Francisco. One last words from Professor Belti that has.
Yeah, Professor Vilaplana, thank you very much for your excellent presentation in a very important topic, a very important field with different approach, engineering approach, biochemical approach, nutritional applications. And then this topic for us in Technological Monterrey is very important, attending our research project in food security and nutrition. Then thank you again for the excellent presentation, and we would like to work with you in the next future, and thank you very much.
Well, thank you, Francisco. Thank you, everyone, for participating in this webinar. Please keep on and check our website for the following webinars that we'll have. And well, thank you so much, everyone. Gracias, Francisco.
Muchísimas gracias. Buenos días.
Bye.
Bye.